Hello my dear friends, welcome back to my YouTube channel. Myself, I am Laukus Jogi. I have completed MSc in Agriculture, Seed Science and Technology, Specialization Seed Science and Technology from HNB GUS Central University, Srinagar, Uttarakhand in 2020. I have got 52 rank in PhD program Seed Science and Technology from Indian Council of Agricultural Research ICR All India Entrance Examination for admission in 2021. Today I am going to tell about the topic what is seed. First, history, second, seed production, third, development. What is seed? A seed is an embryonic plant enclosed in a protective outer covering. The formation of the seed is part of the process of reproduction in seed plants. The spermatophytes, including the gymnosperm and angiosperm plants. Seed are the product of the ripened ovule after fertilization by pollen and some growth within the mother plant. The embryo develops from the zygote and seed coat from the integuments of the ovule. Seeds have been an important development in the reproduction and success of gymnosperm and angiosperm plant relative to more primitive plants such as ferns, mosses, and liverworts, which do not have seed and use water dependent means to propagate themselves. Seeds plant now dominant biological niches on land from forest to grasslands both in hot and cold climates. The term seed also has a general meaning that attendants the above anything that can be sown. Seed potatoes, seed of corn are sunflower seed. In the case of sunflower and corn seed, what is sown is the seed enclosed in a cell or husk, whereas the potato is a tuber. In the angiosperm flowering plants, the ovary ripens to a fruit which contains the seed and serves to disseminate it. Many structures commonly referred to as seed are actually dry fruits. Sunflower seeds are sometimes sold commercially while still enclosed within the hard wall of the fruit, which must be split open the reach the seed. Different group of plants have other modifications. The so-called stone fruits, such as the peach, have a hardened fruit layer. The endocarp fused surrounding the actual seed. Nuts are the one-seeded, hard-celled fruit of some plants with an hyacinth seeds such as acorn or hazelnut. History. The first land plant involved around 468 million years ago. The reproduce using spores. The oldest seed-bearing plant were gymnosperm which had no ovaries to contain the seed, arising sometime during the late Devonian period 416 million to 358 million years ago. From these early gymnosperm seed frames involved during the Carboniferous period 359 to 299 million years ago, they had ovules that were born in in a carpel, which were a group of enclosing branches likely used to protect the developing seeds. Seed production. Seed are produced in several related groups of plants and their manner of production distinguishes the angiosperm enclosed seeds from the gymnosperm. Angiosperm seed are produce in a hard or freshly structure called a fruit that encloses the seed for protection in order to secure healthy growth. Healthy growth. Some fruit have a layer of both hard and freshly material in gymnosperm. No special structure developed to enclose the seeds, which begin their development naked on the bracts of corns. However, the seeds do become covered by the corn scales as they develop in some species of conifer. Seed production in a natural plant population varies widely from year to year in response to weather, varieties, variable insect and disease and internal cycle within the plants themselves over a 20 years period. For example, forests composed of lob- lovely pine and short-lived pine produced from 0 to nearly 5 million sound pine seed per hectare. Over this period, there were 6 bumper, 5 poor, and 9 good seed crops. When evaluated for production of adequate seedling for natural forest reproduction. Development. Angiosperm flowering plant seed consists of three genetically distinct constituents. First, the embryo formed from the zygote. Second, the endosperm, which is normally triploid. Third, the seed coat from tissue derived from the material tissue of the ovule. In angiosperm, two process of seed development begin with double fertilization, which involves the fusion of two male gametes with the egg cell and the central cell to form the primary endosperm and the zygote. Right after fertilization, the zygote is mostly inactive, but the primary endosperm divided rapidly from the endosperm tissues. The tissue become the food the young plant will be consumed until the root have developed after germination. Ovule main article ovule. After fertilization, the ovule develop into the seeds. The ovule consists of a number of components. The funiculus are seed stalk which attach the ovule to the placenta and hence ovary or fruit wall at the pericarp. The nucleus, the nucellus, the remnant of the megasporony regium and main region of the ovule where the megagametophyte develops. The micropyle, a small pore or opening the in the apex of integument of the ovule where the pollen tube usually enter during the process of fertilization. The chalaza, the base of the ovule opposite the micropyle where integument and nucellus are joined together. The shape of ovule that as they develop often affect the final shape of the seeds. Plants generally produce ovule of four shape. The most common shape is called anatropous with a curved shape. Orthotropous ovule are straight with all the part of the ovule lined up in a long row producing an uncovered seeds. Campylotropous ovule have a covered megametophyte, often giving the seeds a tight C-shape. The last ovule shape is called epitropous, where the ovule is partly inverted. The turned back 90 degree on the stack. The funicle or funiculus. In the majority of flowering plants, the zygote first division in is transseverally oriented in regarded to the long axis, and this establishes to plurality of the embryo. The upper or chellegal pole become the main area of growth of embryo, while the lower of micropolar pole produce the stalk-like suspensor that attaches to the micropyle. The suspensor avoids and manufactures nutrients from the endosperm that are used during the embryo growth. Embryo. The main components of the embryo are the cotyledons, the seed leaf attached to the embryonic axis. There may be one monocotyledons or dicotyledons. The cotyledons are also the source of nutrients in the non-endospermic cotyledons, in which case they replace the endosperm and are thick and lethy. In endospermic seed, the cotyledons are thin and peppery. Dicotyledons have the point of attachment opposite one another on the axis. The epicotyle, the embryonic axis above the point of attachment of the cotyledons. The plumule, the tip of the epicotyle, and as the phytherapy Phthiri appearance due to the presence of young leaf perimordia at the apex, and will become the shoot upon germination. The hypocotyl, the embryonic axis below the point of attachment of the cotyledons connecting the epicotyl and the radicle being the stem root transiting zone. The radicle, the basal tip of hypocotyl growing into the primary roots. Monocotyledons plant have two additional structure in the form of sets. The plumule is covered with a coleoptile that from the first leaf, while the radicle is covered the coryza that connects to the primary root and adventitious root from the sides. Here the hypocotyl is a rudimentary axis between radicle and plumule. The seeds of corn are constructed with these structure: pericarp, skeletum, scutellum, single large cotyledon. The absorb nutrient from the endosperm, plumule, radicle, coleoptile, and coryza. These last two structures are sheath-like and enclose the plumule and radicle, acting as a protective covering. Seed coat. Seed coat. Seed coat redirects her for artificial coat seed coating. The maturing ovule undergoes marked charge in the integuments, generally a reducing in disorganization, but occasionally a thickening. The seed coat forms from the two integument or outer layer of cell of the ovule, which derived from tissue from the mother plant and inner integument from the tegmen and the outer from the testa. The seed coat of some monocotyledons plants such as the grasses are not distinct structure but not fused with the fruit wall to form pericarp. The testae of both monocots and dicots of often marked with the patterns and textured marking are have wings of tufts of hair. When the seed coat from forms from only one layer is all it is also called is testa. Though not all such testa and homologous from one species to the next. The funiculus abscess is detached at fixed point. Abscission zone, the scar forming and ovulation depressions. The hilum and anatropous ovule have a portion of the funiculus that is adnate fused to the seed coat and which forms are longitudinal rise or raphe just above the hilum. In bitygamic ovules, Gossypium derived her both inner and outer integuments contribute to the seed coat formation. With continuing maturation, the cell enlarges the outer integuments while the inner epidermis may remain a single layer in may also divided to produce to the three layer and accumulate starch and is referred to as the colorless layer. By contrast, the outer epidermis become teniferous. The linear integument may consist of 8 of 15 layer. As the cell enlarges and starch is deposited in the outer layer of the pigmented zone below the outer epidermis, this zone begins to lignify while the cell of the outer epidermis enlarge radically and their walls thicken with nucleus and cytoplasm compressed into the outer layer. These cells which are borders of their inner surface are called placid cells. In the inner epidermis, the cell also enlarge readily with plate-like thickening of the walls. The mature inner integument has a placid layer of pigmented zone with 15 to 20 layer, while the innermost layer is known as the frigor layer. Gymnosperm. In gymnosperm which do not ovaries the ovule and hence the seed of exposed. This is the basis of their nomenclature. Naked seeded plant to sperm cell transferred from the pollen do not develop or the seed by double fertilization but one sperm nucleus units with the egg nucleus and the other sperm is not used. Sometimes each sperm fertilizes an egg cell and one zygote is then aborted or absorbed during the early development. The seed is composed of the embryo, the result of fertilization and tissue from the mother plant, which is also from a cone around the seed is coniferous plants such as pine and spruce. shape and appearance. A large number of terms used to describe seed shape, many of which are largely self-explanatory, such as bean shape, reniform, resembling is a kidney with lobed end are nether side of the hilum, square of oblong, angular with all sides more or less equal or longer that wide, triangular, three-sided broadest blue middle, rounded of both end and egg-shaped ovule are ovate, broader at one end, big rounded but either symmetrical about the middle of are broader below the middle or broader above the middle. Other less obvious terms include discoid, resembling a disc or blade having both thickness and perial faces and with a rounded margin. Ellipsoid globes are spug lobes but less than spherical, lenticular among avoid reniform and sectoid straight. Seed are straight with parent longitudinal line or rise. The components color are brown or black, one color are frequent. The surface varies of highly polished to considerably rugged. The surface may have a variety appendage, seed coat, and the described by terms such as papillate, digitiform, finger-like. A seed coat with consistency of cork is referred to sub suborezes. Other terms include crustaceous, hard, thin, or brittle. Structure. A typical seed includes two basic parts. First, an embryo, second, seed coats. In addition, the endosperm forms of supply of nutrients from the embryo is most monocotyledons and the endospermic cotyledons, dicotyledons. Seed types. Seeds have been considered to occur in mainly structurally different types. These are based on a number of criteria, of which the dominant one is the embryo to seed size ratio. The reflects to the degree to which the developing cotyledons absorb the nutrients of the endosperm and thus obliterate it. Six types occur amongst monocotyledons, 10 the dicotyledons, and 2 the gymnosperm linear and sepulate. This classification is based on three characteristics: embryo morphology, amount of endosperm, and position of the embryo relative to the endosperm. Embryo. In embryospermic seed, there are two distinct regions inside to the seed coat on upper and larger endosperm and a lower is smaller embryo. The embryo is the fertilized ovule in an immature plant from which a new plant will be grow under proper condition. The embryo has one cotyledon or seed leaf in monocotyledons, two cotyledons in almost all dicotyledons, and two or more in gymnosperm. In the fruit of grains, Caryopsis, the single monocotyledons in seeded shaped and hence called as the scutellum. The scutellum is pressed closely against the endosperm from which it absorbed absorbs food and passes it to the growing parts. Embryo described rods include small strath, bent, crowd, and crulled. Nutrient storage. Within the seed, there usually is a store of nutrient for the seedling that will grow from the embryo. The form of the stored nutrition varies depending on the kind of plant and in angiosperm. In stored food beings as a tissue called endosperm, which is derived from the mother plant and the pollen by double fertilization. It is usually triploid and is rich in oil, starch, and protein. In gymnosperm such as the conifers and food stored tissue, also called endosperm, is part of the female gametophyte, a haploid tissue. The endosperm is surrounded by the aleurone layer filled with proteinous aleurone grains.